going to talk about protein synthesis. Protein synthesis is how your body makes proteins. There are two basic steps to how it works. There is transcription and translation. Transcription starts in the nucleus and it transcribes, which means to copy. So it copies its DNA onto a strand of mRNA. The mRNA then goes to the ribosome where the proteins are made, and that is called translation. Translation is like when you translate something to make sense of it all. So the ribosome translates the mRNA to make the protein.
We're going to talk about the first step in protein synthesis, which is transcription. Transcription starts in the nucleus where it receives a message to make a protein. It starts by the DNA unraveling to expose a certain gene that is responsible for the making of that protein. Here I've made a diagram out of corkboard and styrofoam of the DNA unraveling. As you can see, there are 4 nucleotides. There's thionine, which is its complementary base pair to adenine, and there's guanine, which has a complementary pair to cytosine. Once the two strands have separated, the leading strand is exposed for the mRNA to attach. The mRNA is the messenger ribonucleic acid. It starts by each complementary pair of the messenger RNA attaching to the leading strand until it's at the end of the gene. Once it has attached and is made fully and completely, it will detach, leave the nucleus, and head to the ribosome for the second part of protein synthesis, which is translation. At the very end, the two strands will reattach to be ready for the process to start all over again.
Why do they make mRNA?
mRNA is used as a messenger. Since the DNA is too large to leave the nucleus, it makes mRNA, which is small, and can deliver the message from the nucleus to the ribosome to tell it to make what protein.
Interesting.
The second part of protein synthesis is translation. Translation is much like making cookies. You need somewhere to make it, which is the kitchen, or in the case of protein synthesis, is the ribosome. You need a recipe, which is your mRNA, or your recipe to make the cookies. And you need your ingredients, which are your amino acids, or your cooking ingredients, milk, eggs, and sugar. It starts by the mRNA entering the ribosome to be read. They are read in triplets, or codons. Every codon has a start and a stop codon. Every start codon is AUG. There are 3 different stop codons. I use the stop codon UAA. Every codon has an anticodon to get the amino acid. The anticodon to AUG is UAC. This is the anticodon. As you can see, there is a base triplet as well as the top portion, which is the protein binding site. This is where it gets the amino acids to bring to the mRNA. So each anticodon will bring a specific amino acid associated with it to the mRNA to link through the chain. It'll go on so on and so forth until it reaches the stop codon, telling the chain that it is finished. Once it's at the end, it'll have all the proteins, or all the amino acids, linked together in a chain to form the protein.
The last part of protein synthesis I'm going to talk about are mutations. Mutations are what happen when something went wrong during the making of that protein. There are 2 types. There's base substitution and frame shift mutation. Base substitution is when the entire nucleotide base is removed. This can cause the premature termination of the mRNA during translation. Also, it could cause there to be no start or stop codon, which makes the making of the protein basically impossible. The second part is insertion or deletion. In the original GAU, UCC, GCU equation at the top, which contains the amino acids aspartate, styrene, and alanine, and when you insert something, so say you were to insert U into the equation, it would completely throw off what amino acids you were making. Same with deletion. If you were to add an A to the equation, it would make every single amino acid different. That would cause the protein to not make the same thing.
What is sickle cell anemia?
Sickle cell anemia, how's it caused?
Yeah.
Sickle cell anemia is when the hemoglobin, or the red blood cells, are not formed properly. That could be caused from insertion or deletion. So for insertion, the hemoglobin, the protein that makes it, was not made properly, which made the red blood cell not have the proper base structure.
Oh, okay.